Hi everyone, my name is Cebile Lekhuleni from the National Institute for Communicable Diseases. I'll be touching on the impact of pneumococcal conjugate vaccines on invasive pneumococcal disease-causing lineages in South African children. Just to give a brief of a background, Streptococcus pneumoniae causes both non-invasive and invasive pneumococcal disease, including meningitis and bacteremia. And after the introduction of pneumococcal conjugate vaccines 7 and 13 in 2009 and 11 respectively, we saw a drastic reduction in invasive pneumococcal disease that was caused by the serotypes that are targeted by these vaccines. Subsequent to this, we saw an increase in the prevalence of non-invasive, non-vaccine serotypes. So this brings me to the importance of continued genomic surveillance, not only to advance future vaccine design and policymaking, but also to assess country-specific coverage. Just to give a brief background of our surveillance program, it is a national laboratory-based surveillance that started in 1999. There are over 200 participating laboratories across the country. There is also enhanced surveillance where we have around 30 sentinel sites that submit additional data such as HIV serostatus and patient outcome. So our center at the NICD works on both viral and bacterial respiratory pathogens, and Streptococcus pneumoniae is one of those. And this is just to give a brief overview of what our serotype distribution looks like. So here we have the predominant serotypes, and they are stratified by vaccine period. So you have the pre-PCV period, PCV7, early PCV, and late PCV13 periods. And you can clearly see that after vaccine introduction, there was a decrease in the number of serotypes that are causing disease and those that were targeted by vaccines. And those are highlighted by the asterisks there. This brings me to the aim of my study, which was to determine the genomic population structure of invasive pneumococcal disease isolates before and after the introduction of these vaccines in South Africa. The objectives included evaluating replacement lineages that express both vaccine and non-vaccine serotypes, and we'll see the importance of that later on in the slides. We also aimed at identifying predominant pneumococcal lineages that are causing disease after vaccine introduction. We also looked at antimicrobial resistant traits as well as phenotypes. And last but not least, we looked at the vaccine coverage of higher valency vaccines that are currently under development. Just to give a brief overview of our sample selection, there were over 54,000 reported cases of invasive pneumococcal disease for the period 2005 to 2020. And of those, we ended up sequencing roughly 3,100 isolates down there. And they were representative of the entire collection. And these isolates went through our normal processes of whole genome sequencing. So we cultured from their cryopreserved state all the way through DNA extraction, followed by sequencing and analysis using bioinformatic pipelines. Just to break down a bit of the bioinformatics, we did our sequencing using Illumina platforms, and quality control was done using the Sanger pipeline. Typing, we did SeroBA for in silico serotyping, and just to mention that SeroBA had a 99% concordance with our phenotypic serotyping methods in our laboratory. We used MLS to check for sequence typing. We used POPUNC for GPSC identification. In long, it is global pneumococcal sequencing clusters. And we use the CDC pipeline to identify resistant determinants for penicillin, as well as other AMR genes. For lineage-specific analysis, we used BWA for reference-based mapping. We used Gabbins for identifying recombination blocks. We used RexML for recombination-free phylogenes, since Gabbins mask recombination. We then used MicroReact and Fandango for visualization. And all statistical analyses were carried out in R. So moving on to the results, we identified 155 GPSC lineages, as well as 6 unassigned lineages. So this figure, we've highlighted the 10 most common pneumococcal lineages, and you can clearly see the association with specific lineages as well as their antimicrobial resistant profile. And when looking at the 10 most common serotypes, apart from GPSC3, all of these predominant were associated with vaccine serotypes, and they had at least one resistant determinant. And then looking at the dynamics of the global pneumococcal sequencing clusters, so this figure only included isolates that had over 40 isolates, and then less than that was chucked out. And the solid blocks are showing vaccine serotypes. The hatched blocks are showing non-vaccine serotypes. And we can clearly see the intervention effect, or rather the effect caused by introducing pneumococcal conjugate vaccines, where you can see the drastic decline in serotypes that are targeted by these vaccines when you look at the pre-PCV period, PCV7, early and late PCV period. And for example, when you look at GPSC1, that is highlighted there, there was a significant reduction in disease when comparing the pre-PCV period and the late PCV13 period. Similar patterns were observed for these other lineages, including 5, 8, 10, and so forth. Also, when we were comparing the pre-PCV period with PCV7 period, and also the pre-PCV period with the late PCV13 period, there were also significant reductions in lineages 2, 9, 13, 48, and 77. So now, going on in the slides, we'll look also at a more detailed overview of the effects of pneumococcal conjugate vaccines on these lineages. And we'll look at GPSC5 and GPSC9 as highlighted there. So when you look at GPSC9, this lineage largely expressed vaccine serotypes before the introduction of pneumococcal conjugate vaccines, and those were serotype 23F, 6A, as well as serotype 19F. However, after the introduction of pneumococcal conjugate vaccines, this lineage largely expressed serotype 35B, which is not a vaccine serotype. And it also contributed quite a bit to antimicrobial resistance. So, and the reason for this is you can find that within a lineage, it is expressing both vaccine and non-vaccine serotypes. And when you introduce the vaccine, the non-vaccine serotypes actually occupy the vacated niche of the vaccine serotypes. But the biological background is still the same, so they still harbor the same antimicrobial resistant determinants. Another example would be GPSC9. With GPSC9, it largely expressed vaccine serotype 14, as you can see on the phylogenetic tree there. And after the introduction of pneumococcal conjugate vaccines, it then expressed in larger proportion serotype 15A. And quite interestingly, when we looked at the CPS locus, which codes for these serotypes, there were reasonable variations among these serotypes, although they were part of the same lineage. So this was also an interesting finding, and it also supports findings in the literature of serotype replacement and recombination and all of that. So moving on to antimicrobial resistance. So we saw a large reduction in antimicrobial resistance when you look at the overall collection of our samples. So you can see there were reductions for penicillin, erythromycin, clindamycin, tetracycline, cotrimoxazole multi-drug resistance as well. But when we actually looked at just the non-vaccine serotypes, there were actually some increases in the proportion, or rather the incidence of non-vaccine serotype AMR. So we saw increases in penicillin, erythromycin, and multi-drug resistance. And this is the same concept as with GPSC5 or GPSC9, as I've mentioned in the previous slide, that these serotypes still have the same genetic background as their vaccine type counterparts. And And last but not least, we look at the coverage of the vaccines that are currently under development, and we only focused on the late PCV13 period, as highlighted in gray here, because that's the period we're currently in. And we saw that there was a reasonable addition in protection if we were to introduce PCV20, 24, or 25. And obviously, PCV25 offered a higher proportion of coverage. But additional studies still need to be conducted to evaluate the cost effectiveness of this. Okay. And the added coverage of PCV25 was because this vaccine formulation covers some of the serotypes that are currently predominant in South Africa, and those are the ones that are highlighted there. So in conclusion, we observed significant changes in the circulating lineages in South Africa after the introduction of pneumococcal conjugate vaccines. And although resistance, antimicrobial resistance decreased overall, we saw that there were actually increases in antimicrobial resistance when you're just looking at the non-vaccine serotypes that are currently predominating now. And also, we are saying that higher valency vaccines may have the potential to further reduce IPD, but there's still room for more studies to evaluate how useful this could be. And yay, these are the people that helped me in my study. And I was just looking at the time all the time, and I'm on time, so great. Yeah, any questions?